Hi everyone, we're at the top of the hour, so I'm going to get started. I'd first like to thank the department for hosting Dr. Banker's visit. I also want to thank Teresa, Melissa, Jarrett, and Tracy for helping organize travel and the lunch. It's been really helpful. And a special thank you to Ricardo for bringing back to this entire seminar series. So Dr. Roxanne Baker will be speaking to us today about her work on understanding marine communities by combining mathematical models, fossil records, and food web theory. Dr. Baker received her Bachelor's of Science in Biology with a minor in Chemistry from Denison University. She then moved to the Department of Geology at UC Davis for a PhD with Dr. Herat Vermey. That's cool. Upon completion of her PhD, she held a variety of postdoctoral research positions at UC Davis, the California Academy of Science, and Miami University, which is in Ohio, not Florida. I read that today. She currently is a postdoctoral researcher at the University of Nevada, Las Vegas, in the Department of Geoscience, working with Dr. Carrie Tyler. There will be a happy hour following today's seminar at Bombs Away, so I encourage you all to join us. Folks who can carpool will meet in the lobby 5:05-ish and head out. And with that, I present Dr. Roxanne Baker.
Thank you all so much for being here, and thank you, Cheyenne, for inviting me, and thank you everyone for helping coordinate my travel. It was really easy to get here, which was awesome. So again, today I'll be talking about using community food webs to better understand marine ecology. Can everyone hear me okay? All right. So I just want to quickly thank or acknowledge all of my collaborators and funding sources. My advisor, Carrie Tyler, her graduate student, Madeline S., my collaborators at the Cal Academy, Drs. Peter Ruppenrein and Scott Sampson, and then Ashley Deneen at the University of California Museum of Paleontology. So without all of them, this really wouldn't have been possible. And so without further ado, I'll dive in. And so in my research, I really like to use food web networks and a deep time perspective to better understand the evolution and ecology of ecosystems. And so I think most people in this room will be really familiar with what a food web is, but just in case, a food web is basically a diagram of all of the members of a community. And we usually draw the path or flow of resources or predator-prey relationships as arrows moving from the prey species to the consumer. And so on the left, here's a diagram of an idealized sort of kelp forest ecosystem. And on the right, you'll see a more sort of a schematic example. We can also represent all of these taxa as simple nodes, and then the links between them is the arrows drawn between those nodes. And again, food web networks are a really powerful tool to understand ecosystems because they summarize all of the interactions in a system. And so we know already that ecosystems are not random networks. And on the right, I have shown a real food web reconstructed from a modern Jamaican coral reef. And this was published by Ruppenrein and Deneen in 2018. And if you were to zoom in on one of these edges, it would be composed of hundreds of nodes that represent species in this ecosystem. And when I say that ecosystems are not random networks, all these interactions are highly ordered. One of the things that we tend to see fall out in terms of structure in these networks are modules. So each of these colors and the numbers around the web represent groups of organisms that interact more often with one another than the other organisms in the food web. And so we also know that functional diversity and how richness is apportioned across those functions and how all those functions interact in the food web dictate higher level community properties such as resilience, the ability to sort of be resilient or resist a perturbation, to be stable in the face of that perturbation, or to persist as a species or a system over long periods of time. And we also know, finally, that real ecosystems, ones that we study in nature, perform better in terms of resilience, stability, and persistence than randomly generated food webs. And what I like to do is combine this sort of food web perspective with the deep time perspective And the reason this is important, or the reason I think this is important, is because we really are humans. We study ecosystems over our lifetimes, over human timescales. And unfortunately, all of these systems that we're interacting with took millions of years to evolve into their current state. And so it's really important to start to study these processes over longer timescales to really get at how do ecological communities evolve as a system. And so it's also important to bring in this deep time perspective, because as we're trying to, I've talked with a lot of you guys about this today, but as we're trying to understand and conserve modern systems, we're already looking at a highly degraded version of what was here before. And so we really need to go back in time to look at that pre-human baseline to understand, okay, what did the system really look like? How did it function before humans got here? And without that sort of perspective, it's really difficult to see how far has the ecosystem come. And also by having that other time point in the past, we can really start to forecast ecosystem responses into the future. And so here's a diagram just sort of illustrating this concept of shifting baselines, which is the idea that we just gradually become accustomed to in our lifetime as this trophically downgraded system. So this is what the oceans might have looked like in 1800. By 1950, our parents thought, or my parents thought, this is what was normal. And then by today, I mean, this is really sort of what's standard, maybe go out there and just look in the ocean, which is unfortunate. But we really shouldn't think of this as normal, because this is a very geologically brief state. And so the first sort of case study I'll be talking about today is some work I did looking into the potential effect of stellar sea cow in kelp forest communities. Where was that thing? I must have. Okay, so Bolin et al. Put out this paper in 2021, which was really neat. And they were discussing the different ecological effects that this mega herbivore, now extinct mega herbivore, stellar sea cow, could have contributed to kelp forest ecosystems. And for anyone who's not very familiar, stellar sea cow, again, was this giant mega herbivore that was present hundreds of years ago in these West Coast kelp forest communities. And this was a very large animal. I think it exceeded over 3 meters in length. And these were really, or we think they were really buoyant animals. They were probably feeding a lot on kelp fronds near the surface. They probably weren't able to dive quite a lot based on what we think about their fat content. And so what I was discussing, or what Peter and Scott and I sort of developed out of this, was a specific hypothesis for how stellar sea cow could have affected kelp forest ecosystems. And specifically, we were interested in if they're grazing on fronds at the surface, is that going to cause more light to penetrate to the benthos? Are we going to see an increase in algal abundance? And would that algal pool act as an alternative food source to buffer against the kelp transition into urchin barrens? And so essentially, we developed a mathematical model to try to mimic the effect of the sea cow grazing behavior in the kelp forest, see what effects this had on kelp forest composition and dynamics. And so I think a lot of people here are going to be really familiar with the system. But so what we sort of think of as a healthy forest in the modern are these really dense kelp forests where we have some understory algae around. And our major grazer in the system is someone we're all very familiar with, the purple sea urchin. And in this system, the major predators are the sea otter and then the sunflower sea star, Pycnopodia helianthroides. And so the sea urchin's main source of food is algal detritus, and they'll sort of hide in their crevices, grab food when it comes by. And they're usually kept in their crevices by the predatory behavior of these two critters. And but when predation pressure releases or their food source decreases, they might venture out of their crevices to forage for food. And this is when we get the situation that we see in the modern, these urchin barrens. So again, these urchins are now leaving their crevices. They're storing these kelp holdfasts as a source of food instead of eating any of those other sources. And so this is our schematic diagram representing the major players in this model system. And so here at the center, we have the purple sea urchin. So that's being consumed by the sea otter and the sunflower star. And then the blue arrow denotes a negative effect on that population. So this is sort of the food web relationship and a negative effect on that population. So similar for the sunflower star, the sea urchin is feeding on the algal detritus and the kelps, and then the kelp is the main source of algal detritus in this model. But what we're really interested in is, would this forest have been different in the presence of this mega herbivore stellar sea cow? And so here is what our model looks like when we add the cow. So again, we still have the purple sea urchin being predicted on by the sea otter and the sunflower star. We have the kelp contributing to the algal detritus, but we've also added the understory algae, which is contributing to that algal pool. And then we also have added stellar sea cow, which is feeding on the kelp fronds, which has, kelp fronds and kelps benefit from each other. And then kelp fronds shade out the understory algae. And then finally, we ran, I think in total, 1,600 runs of this model over different perturbation scenarios, which were one final thing. We had natural perturbation of the giant kelp from wave action, and then we test our model under warming scenarios and a sea star, wasting disease scenario, and then a combined perturbation with warming and sea star wasting disease. And so here's all the math to go with it, which you can find in the paper, but I was going to gloss over that for now. And so the punchline is that we do see multiple kinds of forests. It seems like the sea cow does change the relative composition of the kelp to the understory algae. So on the x-axis, we have the categories for our historical and modern systems. On the y-axis, we have the kelp and understory densities. And then, on the right is the historical system. Oh, sorry. On the left is the historical system. On the right is the modern system. Green is kelp, blue is algae. And in the historical system, we have more similar abundances of kelp and understory algae. Whereas in these modern forests in our model runs, we do have a different relative composition of those two primary producers. So it seems like the sea cow in our model runs is really structuring these forests differently. And then also, so on this figure, I'm showing the different perturbation scenarios on the x-axis. And on the y-axis are the frequency of our simulations that were maximally resistant. So when they were subjected to any of these sets of perturbations, they did not undergo a state shift from the kelp forest to the urchin barrens. And so we see that and the final thing is, I forgot to mention this earlier, we have the historical model and the modern model. And then we also modeled both with and without sea otters, because perhaps unsurprisingly, we started to see some really interesting effects of this keystone species. And so what we see is under all of those configurations, they all are very resistant to a warming only scenario. But there is a sea cow effect when we compare the historical and modern models, in that the sea cow does seem to confer some resilience to that critical state shift when the sea cow is present in the disease and warming plus disease scenarios. But it's also interesting that the system is always more resistant with the sea otter present. So there's something, I mean, I think there's something interesting going on there. And I think it just goes to show that, or supports this longstanding idea that the sea otter is a keystone predator, not just in how they control the urchin directly, but it would also appear that they seem to contribute to the system in some indirect ways that we can't quite put a finger on. We have to dig into these a little bit more. All right, so just to summarize that, so could we promote kelp forest resistance collapse by mimicking the function of the sea cow? So first, I'm not promoting the idea that people go out there and start harvesting kelp fronds to promote kelp resilience. But I think there are some interesting results here that suggest, okay, this kind of behavior does alter the composition of the forest and perhaps also the dynamics of the system. But going out and doing this would definitely first require further model verification and definitely very, very careful experiments. More broadly, though, I think this is a really interesting example of how these top consumers, whether it's a very large active herbivore like Steller sea cow or a predator like the sea otter, but powerful consumers have a really outsized effect on community structure and dynamics, and not always in the very direct predator-prey relationships that we would expect. And I think this just really points to the need to characterize past ecosystems. Again, going back to the idea of shifting baselines and how we understand ecosystem functioning, there really is a need to understand the past ecosystem state so we can understand how much it's changed and really start to get into these underlying mechanisms of community dynamics and how that relates to structure. All right, so I'm also going to talk today about, I'm going to walk further back in time and discuss how I've used ancient food web networks to better understand consumer ecology in the fossil record. Okay, so talking to you guys a lot today, I think it's pretty clear that I'm really interested in sort of the relationship among food web structure, biodiversity, and ecosystem dynamics. And one of the other sort of paths I've taken to look into these questions are using reconstructing food web networks from ancient ecosystems to really look at does changing predation intensity and biodiversity correspond with changes in community structure? And the fossil record, again, offers a really unique opportunity to dig into this. So I just wanted to orient everyone in geologic time for anyone who's not familiar. So it turns out Earth is kind of old. It's about 4.6 billion years old. So here I'll have you focus on the left. So geologists, we always start from the bottom and work our way up because that's kind of how rocks are deposited over time. We always write our grocery list from the bottom up. Right, and so back, and 4.5 billion years ago, or 4,500 million years ago. And just to give you an idea of sort of the pace of evolution over evolutionary time, here are a few sort of important events. So we really didn't see our first fossil prokaryote until 540 million years old, million years after the Earth cooled enough for water to precipitate. And it wasn't until millions and millions of years, we did not see our first animals with skeletons until only 540 million years ago. And we saw our first fish shortly after that, followed by our first sharks about 470 million years ago. Our first tetrapods crawled up in the land around 400 million years ago, followed by our dinosaurs, our mammals, and our humans all the way at the tippy top near zero. So I just wanted to illustrate that there's a huge amount of time before life happens, and we're only really talking about this. And then sometimes we're really only talking about that up there. So there's a lot of time left unexplored that we can really learn a lot from. And this is the slice of time I'll be digging in today, the Mesozoic era. And this is a really cool period of time because we see a huge amount of taxonomic and functional diversification in marine taxa during this time. Prior to this, so during the Paleozoic, I'm actually working on another project in Ordovician, which was really dealing with seas without fish. Like we're really dealing with totally different marine ecologies here. And so moving into the Mesozoic is really when we start to see the first hallmarks of the modern ocean ecosystem. And just so you know, I'm not making this up. Here's another figure sort of illustrating this increase in diversity, where we have geologic time on the x-axis. So again, these are just the last 540 million years when you see sort of complex life in the fossil record. And then number of marine families on the y-axis. And so early in the Cambrian, things were pretty low, kind of cruising along at the reasonable level of diversity. We had an unfortunate, oh yeah, it didn't show up, I was born. We had an unfortunate blip here, but then thereafter it was mass extinction. But then thereafter in the Mesozoic, we quickly regained that diversity and then moved well past it. And not only that, but here's another figure illustrating something similar. So again, this is geologic time, and then these bars all represent different predation strategies. And so we had the radiation of shell-breaking fish starting in the Devonian, so over 350 million years ago. But really, we don't have a lot of these other predator styles coming online until the Mesozoic. And this is really, really important. So this includes shell-crushing cephalopods, marine reptiles. These are the marine dinosaurs. Some paleontologists would hate that I said that. Radiation of shell-crushing crustaceans, radiation of sharks and rays, 4th century celluloids, and then also our drilled gastropods. So again, I just want to underscore, we're seeing a big increase in taxonomic and functional diversity during this time. And so to orient you with my communities that I'll be reconstructing today, I obtained fossil data from basically 4 time slices across the Mesozoic. Basically from oldest to youngest, we have the Anesian, the Carnian, the Chthonian, and the Aptian. You don't need to remember those names. I'll have the youngest, oldest things on all the figures where it's pertinent. And so it turns out also that Earth's continents moves, and this is the approximate continental configuration from the Mesozoic. So this is when we had the supercontinent Pangaea, I believe, starting to break up. And the fossil data that I used to reconstruct these food web networks were obtained from this, actually this should have been a little smaller here, but this large shallow marine basin here called the Tethys Sea. And so data for this data collection for this project really looks like obtaining a lot of data from the paleobiology database. And so this is a database where people submit their fossil occurrence data. We can go in and query it and collect basically occurrences from this time in a specific geographic area. Of course, we do not take the raw data. We clean it and verify it. And then we also supplemented those fossil occurrences with museum collections. So, we went to a few different museums in Europe, and we also collected body size data from direct measurement on specimens in museums and also from the literature. And so the first process in food web network reconstruction, it's obviously tough in the fossil record because we can't directly observe how species interact with one another. But we have an approach where we can estimate this and overcome some of that uncertainty with many simulations with a statistical approach. And so what we do is we take each unique species from all of our communities, and we categorize them into ecological guilds based on common traits. So main habitat, diet, we figure out diet from another number of sort of lines of evidence. So morphology, modern analogs, gut contents, bite marks, gig size. And so we group all those taxa into ecological guilds. And then we create a guild level food web or meta network. And so basically we draw food web interactions between all of those ecological guilds. And so here's a figure that sort of shows this process. And so here we might have multiple species grouped into several ecological guilds. And then what we do is we use this guild structure to generate thousands of species level networks for each community that we're trying to reconstruct. And so the way we do this is we randomly draw those links between predators and prey in a way that's consistent with that guild level topology. And so even though we don't know for sure what these food web relationships would have been, by generating thousands of species level networks, we can start to estimate what that looks like. And so on each of these 1,000 species level networks, we take note of the number of species, number of guilds and interactions in each community. And then we also measure for each species the maximum chain length. So what's the longest chain that it's at the end of, and the network trophic position, which I'll discuss on the next slide. Oh. And then we collapse all of these metrics down to each guild over all 1,000 species level networks, and then look at how the distributions of those guild parameters change among the communities to see how the ecology changes. And so just to talk a little bit more about network trophic position, we don't determine network trophic position using discrete levels. We actually calculate it as essentially the average shortest distance of a predator's prey species to the primary producer level. And so here's the equation to calculate that. But we can also think about it graphically. So if you look at the figure on your right, so we might have two taxa, species 1 and 2. And taxon 1 is only feeding on one of their species that's at the end of a long food chain. So even though, they both have a maximum chain length of 1, 2, 3, 4, 1, 2, 3, 4. Taxon 1 is only feeding on that species at the end of the long chain, so it has a high MTP, whereas this guy is also feeding at intermediate levels in the food chain, so it has a lower network trophic position. And so what I'm going to show next is some of our data from this. And what I did was use a Bayesian linear regression to look at the relationship between network trophic position and maximum chain length and compare that among all of our communities. Oh, and what the figure on the next slide shows are about 100 of the 4,000 linear estimates from the algorithm. Oh, that looks less good on the screen. Oh, well, sorry. But I think you can still see it. So on the x-axis, we have maximum chain length. And again, we have those communities from oldest to the youngest, purple to yellow. And on the y-axis, we have network trophic position. And so if we map these out, the slopes pretty clearly fall into two categories. The Bethonian has a distinctly higher slope in the space. And what that's kind of showing us is even though all these communities sort of have a similar maximum chain length range, the Bethonian clearly has higher network trophic position values at the same maximum chain length. Interestingly, so the Carnian it's hard to see in this version of the figure, but the Carnian has a much wider spread, so its relationship in this space is much less well resolved. And then also interestingly, the oldest community and the youngest community have very, very similar slopes. They are statistically the same. So why? So why does the Bethonian have this, the second youngest community have the highest slope? And again, going back to this idea of how we think about food chains, we have taxon 1 and 2, where we can kind of think about more taxa in the Bethonian, that second youngest community are going to be like number 1, where they might have a similar maximum chain length, but based on this line, it really looks like they're feeding more often at the ends of long chains. Whereas in these other 3 communities, they have a similar maximum chain length again, but they're feeding more often at intermediate levels. Yeah, so again, we're seeing that those consumers are just feeding more often at the end of long chains. And overall, I think this is showing that we're seeing less trophic omnivory in the Bethonian community. So what's driving this pattern? So here's a nice figure from Nolan Falls 2016. So again, on the axis, we have geologic time. And on the y-axis on the left, we have strontium, which is a proxy for continental weathering runoff from the continent into the ocean. So it's basically how many nutrients are getting exported from the land into the ocean. And then we also have species diversity for each of these through phytoplankton groups on the right y-axis. And so I think what we're seeing, and we can see that especially the coccolithophores and the dinoflagellates really start increasing in diversity during the Triassic. And so what I think we might be seeing in the Bethonian, which occurs here during the Jurassic, is we're really starting to see a spike in that primary producer, that energetic base in the ecosystem. So what I hypothesized in this paper is that perhaps, okay, we're getting a larger energetic base of the system, we're getting a lot of really voracious predators that are then eating at the ends of those long chains, which might be possible because, okay, we have more primary production here, but then why does that condition then change moving to the Aptian in our youngest community? Because we do see a re-expansion of omnivory in that community. And I think that really has more to do, sorry, I forgot my slides. And it turns out we can start to dig that out when we look at a more modern community. And so the final sort of analysis that I did in this paper was to compare our Mesozoic data to a modern marine community. And so here I'll show you some data comparing those Mesozoic communities to food web data from a modern Jamaican coral reef. And these data were published by Brugnerine and Deneen 2018. And the data were collected over a 50-year period ending in 2011. And just a few things to note, the time period over which these data were collected already represent a very heavily affected system. And if we look at what literature has to say about it, there's evidence that there's already been a 71% decline in shark abundance since the mid-Holocene 5,000 to 7,000 years ago. So if we plot the same type of food web data from the Jamaican reef onto our Mesozoic systems, we see that the Bethonian data, the second youngest Mesozoic community, is not unique. And it turns out that the cause of that lower slope in the Aptian, youngest community, was due to organisms like sharks and large predatory fish, which sort of were responsible for the higher proportion of shorter food chains, lowering that line. And we know that in modern ecosystems, such as coral reefs, sharks, even though we kind of think of them as this bad apex predator that always feeds on the end of long food chains, they are really important intermediate predators. And so again, we're seeing that these large sharks have been extirpated from this modern community. And I should say too, the predators in this Bethonian community are these big Mesozoic marine reptiles that were feeding on other very large sort of animals like that. And so given this recent shark extirpation, how would we expect a pre-human slope of this, these parameters to look like for the Jamaican Reef? Would the addition of large consumers, like sharks, if we restored that shark diversity, would that drop the line? Would we reestablish some of those shorter food chains? Or would it still look like the Bethonians? We need to look at something else that's controlling food web structure. But in any case, I think that this does again underscore the importance of how do different consumers with different ecologies modify the structure of these food web networks. And so I just want to sum this up here that, you know, whether we're talking about health forest ecosystems and the role of stellar sea count altering community composition, which might ultimately lead to resilience, these top consumers really do have an important effect in determining or structuring communities, which has an important role in the way these different functional entities interact and ultimately does affect community dynamics. And I did want to say, although the results I show you from the Mesozoic work so far really focused on structure, one of the areas I'm excited to delve into is, do these changes in structure correspond to changes in stability? So that's one more area of modeling I'm excited to delve into. One other really interesting, I think, result from the Mesozoic work is previous workers on food web networks have suggested that there was sort of a community structure set in place 540 million years ago. And I think that we see a great deal of variation in the structure of food webs over the Mesozoic. So not only is, you know, community structural change associated with, or it's associated with all these big diversification events over geologic time. And finally, and overall, you know, whether we're talking about either of these case studies, I think we do see a really important role for top consumer activity, how it affects community composition, and ultimately we can really start to understand these processes better when we bring in a deep time component to evaluate past ecosystem states and then the processes that have caused them to evolve over geologic time. And so, yeah, with that, I will end and ask for any questions. Yeah.
Thank you very much. If you were to like expand your study to including like say like 15 million years or something into the Cenozoic, right? So inclusive of the end Cretaceous mass extinction, what do you like hypothesize kind of about consumer activity, trophic cascades, that kind of stuff?
Yeah, that's a really good question. So I guess I'll start with saying we do have data from the Maastrichtian, which is the period of time right before the mass extinction event. So I have that data ready to analyze. I don't know what it looks like yet. But I guess how would I expect it to look different in Cenozoic? That's a really good question. Yeah, I would think that it would depend on the environment. I think that,
because on land, the Cenozoic took out the top consumers, and what we saw, you know, kind of trickle down effect that had. So, like, how would that impact those type of community dynamics in the ocean?
Yeah, I think you start to see similar things, especially, you know, when mass extinctions do selectively hit those higher level consumers first. But yeah, I think, sorry, can you repeat your question? I'm not sure if I'm getting it quite right.
The second one or the first?
Sorry, I lost my train of thought.
Oh, I was just saying, well, we know the in Cretaceous extinction obviously, wiped out the dinosaurs, non-avian dinosaurs. And so it took out the top consumers on land, leading to trickle down effects, and how would that match up what happened in the ocean?
I think it would be pretty similar. I think you'd start to see, I don't know if it would be similar. It would depend on the extinction selectivity in the community you were reconstructing, because I could see how if you were just wiped out a lot of the vertebrates that sort of, connect the food web a lot more, then you're kind of maybe going to see something more like the Thungian. But, if you're taking out, I think it could be really different. I'm not entirely sure, to be honest. I think it's a good question though, and something I would definitely like to dig into more. Questions?
You were talking about the effects of the sea cow and its canopy munching. And I wonder, we talked about like, let's not do any experiments yet, but I don't know if you know, or maybe other people in this room know, there are places where kelp canopy is harvested. And I wonder if you could look and see if those places were more or less resilient to urchin barrens.
Yeah, that I think is up next on the like list of questions we want to look into, because I know particularly in Japan there's a great deal of kelp harvesting. I don't know where else it's done a lot, but that was sort of the next thing we want to start talking about. I think that Peter and Scott have some folks at the Nature Conservancy that are talking to about that, but I agree. That's great. Next step.
Is also on that stellar sea cow project. You mentioned the effect of sea otters being important in both the perturbation systems in both historical and modern. Do you have like intuition for what those effects might be? Is it something of the stellar sea cow is allowing more understory algae to grow, and that's giving urchins an extra food resources to consume, and otters are kind of just still doing the same thing? Or is there maybe something else that's not, that you might have intuition for, it's not quite there?
Yeah, I think that's a really good question. I'm not completely sure, to be honest. I think that was you guys are asking really good questions of basically all of the next steps, and I don't have good intuition offhand. I'm really excited to dig into it. Yeah. Thank you.
You mentioned earlier that you're interested in starting a new project looking at the Ordovician time period.
It's already begun.
Or is, as I understand it, one of the major mass extinction events did take place at the end of the Ordovician.
Yeah.
Are you looking at the change in ecosystem food chains from the Ordovician going into Silurian possibly?
So terrific question, and the answer is no. But what we're looking at specifically during that time, and I believe that our time period is sort of middle late. So I think we're starting to see the late Ordovician extinction in our system, but at the very beginning. But we're really focused on reconstructing the food web networks across the biological invasion that we see. So in the rocks that are now Ohio, there was a shallow enclosed basin that was exposed to invaders during periods of sea level rise. And so we see like 3 pulses of invasion. So we're really interested in how does community food web structure change with biological invasion, how does that play out over evolutionary time? But I believe we don't address the extinction. Yeah, it's a great question.
Well, thank you so much.
Thank you very much.
Thank you very much. Reminder,